Debnath sir, you're connected?
There is a buzz. Yes, yes. Okay. There was a buzz of some kind. Yes, I'm connected, madam.
Yes, okay. Good afternoon, sir.
Yes, good afternoon, madam.
Debnath sir, can we start?
Yes, madam. We can start. In between, I'll take 5 minutes just break. Well, if you permit me, and then I'll just get back. So, madam,
Yeah,
you can
yeah.
start from your half. Okay, madam.
Yeah, yeah. Okay, sir. Thank you so much. Over to you, Vijaya.
Yes, ma'am. A warm good afternoon to all. We are now in the session 3 of day 3 of ATAL sponsored FDP on Pharmaceutical Quality System for Product Lifecycle Management organized by Bharatiya Vidyapit College of Pharmacy, Kolhapur. It gives me immense pleasure to introduce you all to our eminent speaker, Dr. Surajit Debnath sir. Dr. Debnath sir currently is the head of Department of Medical Laboratory Technology, Women's Polytechnics, Tripura, with more than about 13 years of experience in biomedical research and high level teaching. Dr. Debnath sir participated in various academic works in institutions of Germany, Denmark, and Hong Kong, besides in premier Indian labs such as AIIMS, IIT Kharagpur, IIT Guwahati, and NBPGR in plant genomics. Dr. Debnath sir is the recipient of Young Scientist Award for different agencies like DBT, DST, and CSIR, and have also received the Tata Trust grant from CME course in the Royal College of Pathologists, London. Sir is the reviewer of several Scopus and Science uh, indexed journals published by Elsevier, Springer, and Biomed Centers. Along with the regular academic activities, he is also engaged with several biotechnological companies in the northern and western India as a consultant. Sir has conducted various workshops and talks acted as a session chair in various international conferences sponsored by national and international academic agencies. Currently, Sir is guiding the guiding group of international students for their internship from the Russia, Turkey, and West African countries. Dr. Debnath sir has filed about 2 patents in 2020-21 in India on biotechnology. With this brief introduction, on behalf of Bharati Vidyapit College of Pharmacy, Kolhapur, we once again welcome you, sir. Now, I kindly request Dr. Surajit Debnath sir to proceed with his session on Peptide Therapeutics hands-on stimulation session on enhancing their stability using machine learning. Over to you, sir.
Welcome, Debnath sir.
Uh, I have shared my screen. I hope it will be visible right away, and we'll start.
Yes, sir. Yeah. Screen is visible, sir.
Hello.
Yes, sir.
Hello.
Yes, sir. Hello, Debnath sir.
Madam, there is a bit technical glitch. I am audible, I guess, madam. Madam, give me some credentials so that I can share my screen. Okay, fine. What? Uh, Hello, Nila madam.
Yes, sir.
My
Debnath
screen
sir.
was visible. My screen was
Yeah,
visible.
your PPT was
the
visible, but
PPT.
it has, yes, PPT was visible, but now we
Mm-hmm.
cannot, yes, now it is visible, sir.
Okay, okay, okay.
Yes.
So what has
Yes.
happened is thank you, the organizers. I'm very much thankful that you have given me the opportunity to interact. And ATAL is also a kind of innovation by that AICT is given us a platform where an interdisciplinary discussion can be done with a variety of different classes of participants who are teachers by themselves. So it is definitely a platform that is developed by teachers for the teachers and with the teachers. So I'm also thankful to AICTE for providing the platform. So the topic of discussion today is peptide therapeutics hands-on simulation on enhancing their stability using machine learning. So what has happened is, as you are all, madam, most of the participants belong to which kind of disciplines, madam?
Sir, majority, I mean, almost 99% participants are from the pharmacy background, and very few, hardly
Pharmacy
three,
back.
four, 3, 4 participants from the engineering background.
Okay, okay. Pharmacy background, absolutely.
Yeah, all are
So
pharmacy background.
they are okay, okay. So they all deal with a lot of therapeutics, and among the
Yes.
therapeutics, there are therapeutics which are peptide based in nature. So what happens is, as all of you will understand, that eventually what will happen is, when we talk about pharmacy, it's again a very large discipline where you are not only doing about salts and APIs, there are peptide based therapeutics as well. A very common and very well known example would be like insulin, the therapy that is a kind of wonder drug and was a blockbuster drug. And for companies, it's a very big profitable business to produce this particular thing. And insulin is a very good example of this to tackle what do you call is this thing, what do you call is the diabetes. Just hold on.
Is there any issue, sir? Debnath, sir, is there any problem?
Hello, am I audible?
Yes, sir. You're audible, sir.
Okay. So, and the screen is visible too, right, madam?
Yes, sir.
Okay. So, as I have said that the companies like Eli Lilly, the Novo Nordics do a lot of business in throughout the world by developing this kind of therapy. So, other than that, there are plenty of other examples as well. So, let us go into the details. So, when we are talking about peptide therapeutics, we are essentially talking generally about three different things. One is diagnostics, then we are having therapy, and lastly but not the least, the drug delivery mechanism that are being developed by using peptide therapeutics. So when we are talking about diagnostics, the most important thing that are we are having is diagnostics through bioimaging, where we are essentially talking about fluorescent tagged molecules, and then we are also having kind of biosensors. Again, if we talk about the sensors that will detect the blood glucose level in a domestic setup, also uses a lot of peptides which are present in the electrodes. Usually these are redox oxidoreductase kind of enzymes which will bring about a particular kind of reaction, and eventually you will be able to understand how much blood glucose is there in the patient, and accordingly the clinician can adjust the dosage. So this is one. And then when we are talking about therapy, there are plenty, but the major what we call aspects that we are interested in is the regenerative medicine, where there are a lot of peptide therapeutics that are being used. I'll give some specific this thing what we call as examples as well. Then we are having cancer treatment, and then antibacterial agents. So we are not specifically calling it antibacterial. Let us call it antimicrobial agents. Then the third topic, which is drug delivery. There are gene delivery mechanisms, some medicine delivery mechanisms as well that has been developed by using peptide. So one more discipline that has been like been in talk, that is Therapeutics, where we are combining therapeutics and diagnostics both in a single entity, and therefore the things can be brought about, which will eventually. Hello, am I audible?
Yes, sir. Audible.
Hello.
But yes, sir. Yes, sir. Debnath, sir.
Audible, but
Yes, sir. You're audible, but I think there is a bit disturbance in the audio from your
no,
side.
I think disruption from some participant side it may be. If you mute rest of them, so I do not. I think there will be this thing minimization of that. Anyway,
Yeah.
so what happens is this is the initial thing that we are having. Then eventually, what we can do is we can talk about these things. These are some specific examples where. Okay. Window. On the tip. Okay. So what has happened is there are some specific examples. If we say engineered antibodies, now this is very interesting. If you see, so and then engineered antibodies. Example which interest pharmaceutical industry is the rituximab. Rituximab is a monoclonal and developed as a therapy for autoimmune conditions like SLE, systemic lupus erythematosus, but eventually it has also been given for in patients like rheumatic arthritis, and then in kids also these are being used with nephrotic syndromes and all such things. And there are one more very good example, Herceptin, which is again a monoclonal antibody and usually given for treatment of breast cancer in females. So other than that, there are several what we call is engineered peptide entities. Let us give some examples of interleukins for cancer immunotherapy. Then eventually we are having EPO, erythropoietin, which is of much use in CKD patients who are already this thing and got rid of their renal system and they are on dialysis, and many a times it may happen that they develop an abnormality in blood maturation because the EPO, erythropoietin, which is produced in the kidney, are being removed, and therefore they require a lot of erythropoietin that is delivered exogenously. So this is also a very good therapeutic peptide which is being used these days. And then there are some more examples, but one example that we should definitely discuss is interferon gamma. Interferon gamma has been like a treatment for hepatDNA viruses. So the hepatitis B, C kind of treatment, terminal treatment of these hepatDNA viruses. They use a lot of interferon gamma, and CGI Chandigarh, that is PG Institute Chandigarh, is the pioneer in using interferon gamma for hepatDNA virus treatment in India, and they have done excellent work over the period of time. So there are several examples where peptide-based therapeutics are in use, and there is a lot of demand, and the pharmaceutical companies are exploring. But there is a problem. Now the problem here is with the shelf life of the compound. So as you can all understand that it will be of much use if a particular drug is of extended shelf life. Understood. So if extended shelf life you are talking about, then you are talking about this thing, what you call is expiry date. So if you are putting your insulin and you need to preserve it in a particular way, and then also there is a particular kind of expiry date. So you should have an idea that how to enhance the so what you call is the shelf life of this particular drug. And in order to do so, protein engineering will give an excellent opportunity to explore. And many development has already been generated, but there are a lot many things that are yet to be done. Other than that, there are several other examples. Coordinator, madam, if the slides are changing in the perfect sync, is it changing or sometimes it is getting stuck?
Everything yes, sir, it is changing.
Okay, fine. So what has happened is there are some other specific examples as well. These are a little bit advanced applications and not very much common, and many are in lab scale. So we'll not particularly talk about it. So one such example I can give is ADCC enhancing antibodies. So antibody dependent cell-mediated cytotoxicity that has been explored for cancer immunotherapy recently, and there has been very good results as well. So this discussion is only to give our participants an idea that in peptide therapeutics field also there is a lot more scope that pharmaceutical technologists and scientists like you can collaborate with biomedical labs like ours, and then we can generate intellectual property, and not in the only in the form of research papers, but eventually which may turn into a patent also. So what happens is and one more very interesting and very useful group of peptide therapeutics is definitely enzymes and catalytic proteins. So all of you will definitely agree this is also a very big market, enzymes and catalytic proteins. For example, in Indian context, dyspepsia and what do you call is malabsorption syndromes are very common in India. And any medicine shop you go, definitely you will get many preparations of enzymes, mainly pepsins and amylase, which comes handy, and it is useful. Many times these are also OTC products. Some of these has become over the year because the demand is so high that without any you are having your prophylactic uses also for this kind of therapeutic things. So there is a huge market, and these also are peptide based therapeutics also. For example, like some specific cases are also there. For example, patients suffering from this what we call as cystic fibrosis. So they do not able to prepare lipase in their pancreas, and eventually they require exogenous lipase. So this is a little special case, but rest of the thing, as I have said, amylase and this what we call is pepsin is very common. These are also peptide-based therapeutics which are being used by the pharmaceutical industry. So here also, if you prepare a amylase preparation by a solid state fermentation through an fungi that you can do, and it is therefore very cheap also. These preparations are also very cheap. 100 ml comes with approximately less than 100 rupees per bottle of a syrup. So it is also very useful, and it gives a lot of this thing in Indian context, because our food habit and rest of the other things are not very are not very superior, and therefore a lack of digestion and things like that are there. So what happens, it is also a very good opportunity that we can enhance the stability of these peptides as well, which will eventually give you an opportunity to explore the market. Companies like Abbott and several other has explored this opportunity to a very significant extent and very much profitable these days. So one more example that I have said is lipase. Now lipase, you cannot make it through solid state fermentation of an enzyme, which is very common in for example, Pseudomonas syringe. If you culture in a solid state fermentation, you will end up with a lot of amylase. Okay, because the fungal hyphae will produce a lot of amylase in order to disintegrate the raw material that is there. The fungi is growing, but lipase is not like that, and you have to use recombinant DNA technology in order to produce it and purify it and do the other things. So the cost becomes a little bit more. So here also they have done excellent innovations. I shall discuss this in coming shortly. So this is the one more example of peptide-based therapeutics are vaccines. Okay, so I hope my screen is visible, and this screen depicts the production of a vaccine, a classical vaccine, which takes about 10 years. Okay, so from ideation to ultimately putting it after the phase 1, phase 2, phase 3 clinical to put it into exactly in the market, it takes approximately 10. But recently you have seen that in COVID period, Indian and several other companies have generated the vaccine within a fraction of 1 year, and sometimes less than that. So how exactly this thing has been possible? This has been possible because we have developed a very good simulation strategy using machine learning and artificial intelligence, which is nothing but using of microprocessor and computer technology for reduction of the false positive and false negative results, which eventually gives you a lot of scope to develop a product in a very rapidly and very small period of time. So we will do a little hands-on. I am not very sure that the participants will be happy or not, but I'll try to put enough this thing so that some participants are also aware of it, and I hope you will also this thing what do you call participates. It's a very small hands-on. So what happens is so this is this thing that if we use machine learning capabilities to a significant extent, we can definitely do valuable research in this particular domain. So this was the previous story of a particular kind of vaccine, which took approximately 1 year to develop. Oh, sorry, 10 years, a decade to develop. But today we can do it within a single year itself. So okay, so this is one more very interesting this thing, that what are the different shall I say considerations before developing a peptide therapeutics. So these are some of the considerations that we should have, that it should have very good biocompatibility, and there should not be much of antigenic, what do you call this thing, that contraindications, like if you are doing developing some kind of drug that is should be delivered through intramuscular or intradermal. Okay, 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 ठीक है एकदम ठीक जगह पे आ गया. Okay, हाँ यही. So what has happened is okay. So give me just 2 minutes, madam. I'll just get back.
Okay, sir.
So, madam, may I please request you to play the first video, madam? Madam, am I audible?
Yes, yes, sir.
Yes.
Okay, okay. So, if you play the video, it is just because that it is a post-lunch session and it becomes very challenging for the speaker to draw the attention of the participants because they already feel dizzy and it becomes a burden. So the video may change the mood. So may I please request our coordinator to
Yes, sir, we
share
are
the
doing.
screen? Okay, fine. Thank you.
So I start the video, sir.
Play
I'm
the
starting
video.
the video.
Yes, in mute condition.
Production of insulin through genetic engineering.
Mute condition. Hello.
Yes, sir. Yes, it's muted now.
In mute condition. Mute. Yes, yes. Go ahead. And many windows you have kept open, so your band ideas will go. So whenever it is required, if you play and open, it will be much better. Anyway, so
Okay,
what
sir.
has happened is this is the story. This yeah. So what has happened is eventually. Initially, what has happened is those patients who are suffering from mainly from various complications of hyperglycemia, they used to given pig insulin. You will be knowing. Sir, pause for a second. So what has happened in pig insulin? There was nothing wrong with that. Only one part of it had a particular kind of amino acid which is not very native to human being. It belonged to the pigs or the swine community. So what has happened is many patients developed long-term complications like what you call is severe kinds of allergy and kind of like organ and things like that. So eventually it was been understood that to isolate swine insulin and then put it into the human patients will do no work, and eventually we have to isolate insulin from human subjects, which is again not very possible. So you cannot sacrifice a human to procure insulin from his pancreas. So what is the way out? The way out is this. Hello. So what was the way out? Am I audible, madam?
Yes, yes.
So the way out is to put in perspective that the human protein sequence is conserved, and then eventually we put it into a host system in order to produce a recombinant insulin, which will purify and eventually inject with the what you call is the patient, and the patient will get rid of the symptoms of uncontrolled hyperglycemia, which the drugs cannot like do any this thing. For example, metformin and glipizide, glibenclamide, these are some kind of sulfonylurea drugs which are being used. But many a times it has been seen that eventually you become resistant to this kind of drugs, and ultimately insulin therapy should be initiated. So in that case, how exactly they do it? Mm, So in this way, what they do is you yes sir, please play. Okay. So these are the blue cells, which are a specific kind of cell which produces the insulin. Okay. So what we do is we'll take the genome out of this particular cell, and then we'll engineer it. Okay. So we'll take the whole genome from this particular cell, and then I isolate a particular area of the whole gene which codes for the insulin, and insulin is a peptide. So in order to do so, what exactly we do is this is the whole genome. Although it doesn't look like this, but they are in the video, they are saying that this is the whole genome. Okay, so the whole genome is present in complex with histones, which ultimately becomes the chrome. So what happens is this the whole genome will be cut down at a specific site, which will give us the exact gene sequence of human insulin. What we'll do is we will put it into a vector. The vector is nothing but a vehicle which will eventually help us to produce the protein. Now this is in DNA, then it will become an RNA and eventually a protein. So that protein we are interested in. Now in order to make it a protein which is exactly like human insulin, you need to isolate this gene. And as I have said, these are the basic molecular techniques that are involved in recombinant DNA technology. Putting the thing in a vector and then sealing the ends by a particular kind of enzyme. There are many other things which is involved. It is not as simple as the video is been shown. Anyway, so later on we will put it into a host cell and which will produce the protein for you. You just purify it through some kind of purification techniques. For example, salt extraction you can do. You can even do lyophilization, then you can do dialysis, and sometimes very good analytical tools you can use like HPLC and affinity chromatography in order to purify the protein. So eventually you end up with a lot of insulin, and which you can bottle and put it to your shelf to sell it to the patients. Okay. Okay, sir. Thank you. So what has happened? There is a limitation to it. Excuse me. The limitation is it is having a particular kind of expiry date, and you also need to preserve the protein in a particular way, isn't it? For all kinds of salts, also you are having expiry dates. But usually you do not need to preserve it in a particular freezer kind of thing in order to decrease its instability. But these peptides are highly unstable by themselves, and therefore we can do a lot more engineering to make them a bit more stable. So how exactly we'll do it? So once again, I'll share my screen. Okay.
Hello, Debnath sir, is there any issue? We can see your presentation. It is visible.
So this antigenicity is very important. But you can also ask me that if it is a vaccine, what will happen? So in vaccine, the this thing is completely different, because when you develop a vaccine, you try enhance the system. Okay. You try to enhance the immune system, and you wish that it really acts like an antigen and not like something which is already there in the system. So there are different perspective to it, that it entirely depends which kind of therapy and which kind of application is this particular drug you are using. And there is one more issue of cross signaling. So there are many, but I'm only focusing on those because many participants may not have specific interest to this particular thing, but it becomes this thing. We can at least have a very basic understanding of these concepts. Then one more thing is cross signaling. Now, what is that? When we're talking about cross signaling, we are essentially talking about some molecular signals. Now, what exactly are these? Let me again give you some examples which will be very common to this particular fraternity of what do you call is the pharmaceutical scientists. So here you will also able to this thing also a very good what do you call is innovation that not recently I would say which is already in the market. The many geriatric patients you will agree that they develop bed sores. Okay, so bed sores are nothing but pressure ulcers. For example, some geriatric patient, an older patient who is bedridden for a couple of weeks. So what will happen on his or her skin? There will be some this thing ulcer development. Now, why is this ulcer develops? Because the skin is constantly in touch with a particular substrate, and in those pressure points, blood circulation stops, and the tissue becomes not very functional. So this is how a bed sore is developed in a patient. So in order to treat bed sore, there is an excellent innovation that the pharmaceutical industry has done. A medicine that is called Placentex. If you go to a shop and ask for a Placentex, they'll give you a gel kind of thing. A very good innovation in peptide engineering. So it is nothing but a homogenate of animal placenta. Okay. So what they do is they accumulate the placenta and then homogenate it by a tissue homogenizer and then put it into a what do you call is this inert matrix of a hydrogel. So what happens is the stability of the peptide, because as you know that in the placenta there are multiple types of growth factors. Okay, so why these growth factors? Because these growth factors will repair the placenta and develop the whole organism out of a single what you call embryo, and therefore a lot of growth factors are there in the placenta. For example, epidermal growth factor, for example, fibroblast growth factor, for example, lot many which are very important to develop muscles, then tissues, then skin, and so on. So what the pharmaceutical company did was they isolated the placenta from a diseased animal, and then they homogenated it in a tissue mixer. It's nothing but a grinder. So after grinding the whole tissue, they'll put some preservative so that it do not get rot, and they remove the collagen, which is nothing but the filler material present in between the tissue, and then they'll put it into a hydrogel, which is an inert matrix and which will preserve the activity of these growth factor proteins to a prolonged period. And then they made into a gel and put it into a bottle by a particular kind of bottling method, and then they will sell it in the market with a brand name Placentex. So what exactly they are selling? They are selling a particular kind of growth factors. Now, if you want to isolate the growth factor in its pure form, it will become very costly. If you go to the market and ask for Placentex cream, it will be mostly 400 rupees. Now, if you want to procure pure form of fibroblast growth factor, which will repair the skin of the patient who is lying on the bed for a prolonged period of time and has developed some skin ulcers, if you want to heal the ulcers instantly, you need to give him fibroblast growth factor and lot many kind of like a cocktail of several growth factor peptides, and your job becomes very tedious, and the patient compliance will also be less because it is highly costly. So what this company have done, they have not purified it as such, but they have made a complex out of it and then extended the shelf life by using this thing what they call is hydrogel and eventually marketing the product with very good success, isn't it? So this is also a strategy that you can do for enhancing the stability of the peptide and ensure better patient compliance. That I'm sure is also the goal of your fraternity of pharmaceutical industry, isn't it? So there are several other issues that should be taken care of while designing a peptide therapeutic. And later on, these are some examples where a very good outcome that has been generated in this particular domain, where they have developed very good quality of peptide therapeutics by developing some specific strategy that we call protein engineering, and by putting what do you call is mutant varieties of enzymes. So these are some common examples where most of the work has been done in Scandinavian countries, including Denmark, where a lot patent has been filed in terms of engineered proteins of various kinds of use that we have included, and several more which have not come across our discussion today. So what has happened is this Novo Nordisk is a very big famous pharmaceutical company with special interest in peptide-based therapeutics. So they have developed the first human insulin also, and now they are developing lot many more. So what has happened is this particular kind of area, the Western Europe has been very active. Then there is Japan and then US, who are also very active in protein engineering for pharmaceutical and other usage. So this is a very simple this thing, a representation of catalytic proteins that we have spoken about. How exactly these proteins work and why these are important for purposes, because they reduce the what you call as the free energy that is needed to produce a product, and therefore these are of huge in demand in various other industries, including pharmaceuticals. So many teachers will find it very useful to teach their students, because at the end of the day we all are teachers and primary job is to teach our undergraduate students. So how exactly you will develop what you call a stability of the peptide? Okay, this is also very important, as some we have already spoken about. One is delivery mechanism. What is that delivery mechanism? Delivery mechanism is, as I have given the example, placentex, where you are putting it into an hydrogel and stabilizing the protein, and also you are reducing the need for complete purification, isn't it? From the placenta tissue, if you want to purify fibroblast growth factor, you need to write a project to AICT for approximately 50 lakhs. After 50 lakhs, you will develop a strategy to isolate fibroblast growth factor and purify it. But what is the use? Because the purification technique will be so costly that if you put it in the market, nobody will buy it, because they have a cheaper variety of this placentex. Okay, but it is also true that there are several other applications of this. Let me tell you some specific examples. For example, if a patient is undergoing a angioplasty, in that point of time, in angioplasty, what they'll do is they'll cut a particular kind of vein and then put it into the heart near the coronary. So what will happen is it will take some time to recover the patient, because a certain area of a particular blood vessel has been removed, isn't it? So the patient needs prolonged what do you call hospital care, which is also very costly today in India. So in order to repair the wound, they need purified versions of this fibroblast growth factor, epidermal growth factor, and again a complex kind of delivery mechanism strategy has been developed. So these are there. Therefore, if you develop a delivery mechanism nicely, also it's a very good this thing innovation by itself. Then there are strategies of immobilization. Okay. So immobilization is, as I have told you about the case of lipase. Now lipase is comparatively less stable than that of pepsin and amylase. Okay. Now this is also the reason that you cannot develop lipase by solid state ferment. Okay, you have to do genetic engineering for that. But rest of the other peptides are comparatively more stable than that of pepsin. How you will stabilize pepsin? So for this also, Abbott has done an excellent work. So what they did is they entrapped the lipase in a silica matrix which is alkaline in nature. Okay. So what has happened is the lipase becomes immobilized, and whenever it enters into the gut of the patient, then only it becomes, it gives a sustained release kind of activity, and eventually it helps to digest uh, fats and fat soluble vitamins, okay, by proper emulsification of fat, and eventually do the thing what it is needed to be. So these are some strategies to stabilize the peptide. Then one more strategy is to assembly of the peptides. Now assembly is a detailed thing that we can discuss, but as our target audience today belong to a particular kind of this thing, so it is a bit lengthy process to discuss. However, we will touch all these specific topics. So in peptide assembly, what happens is we eventually increase the overall molecular weight of the peptide. So I'll give you a specific example where we have isolated in our lab a particular kind of peptide which is very good antipathogenic. So we have analyzed the antipathogenic work and eventually we put it into a text also, which has been published. But later on we found that the stability of the peptide is so low that we cannot make a very good use of it. So in order to enhance the stability, we explored what is the possible strategies that are there, and we eventually found that if we artificially increase the molecular weight, so what will happen is whenever there is a protein, it comes in contact with several other what we call is natural forces like van der Waals, then ionic forces, then hydrophobic interactions, and so many other things. So what happens? The peptide remains very unstable due to the various forces of the nature. So if you can increase the overall molecular weight, and there will be a steric hindrance kind of activity. Now, steric hindrance means you are protecting your core area by putting some artificial exterior domains. Okay. Isn't it? For example, the environment rapidly changes pH. So what will happen? The protein will rapidly degrade. So the degradation takes place on the exterior and the core remains stable, isn't it? So this is a strategy that has been developed and been used for many this thing. So these are the things that we can do to enhance the overall stability. So these are again by means of delivery mechanism. So again, I'll give you some specific examples. I hope that these are of interest to you. So there is a very good case of this. What do you call is this thing? This thing 3-hydroxy cholecalciferol or vitamin D. So this is not a peptide, but its stability is very low. So vitamin D is also a very big business in India. I do not know whether you know or not. You will definitely this thing that nutraceuticals is a big business in India, like developing countries like India, where people have recently developed much more capacity to purchase of this thing, what do you call is lifestyle products. So vitamin D capsules have become like lifestyle products, and they'll procure it very significantly. There's a huge, but there is a problem with conventional vitamin D therapy. Now, what is the problem? The problem is that it is inherently unstable. Okay, and therefore, whenever you purchase a vitamin D calcium tablet, okay, so you will find that on the blister pack they have written the 60,000 international unit of vitamin D with elemental calcium. Okay, so many other things will be there. So what happens is you, while producing the preparation, you give a lot more international units, that is units of vitamin D. But eventually, when it enters into the system of the patient after so many days, the functional vitamin D is very less. Okay, so you ultimately cannot able to give the availability is less than 60,000 international units. Understood. So you need to have a prolonged therapy, and therefore the what do you call the anticipated benefit is also very late to develop. So these are many problems. These are there. But again, here Eli Lilly and company has developed a strategy to generate this particular, to enhance the stability of this particular peptide through a mechanism. What was the mechanism? The mechanism is to put it into a liposome. So what is the liposome? Liposome is very useful and very user friendly and very what shall I say, very nicely it will dissolve into human skin. So what they did is they isolated vitamin D from salmon fish. Okay, salmon is a fish which is present in like not very common in India, but in like nowadays everything is common. But it is basically a fish from a temperate region like Europe, some parts of Germany, and this kind of regions. Okay. So what happens is salmon, a similar kind of variety of fish is available in India that is called hilsa, which you will find plenty in Bay of Bengal and the regions like this. And it is also very famous because I belong to this particular region and I am very much fond of this. So what happens is you isolate salmon cholecalciferol and then put it into a liposome and make it into a nasal spray. Okay. I'm sure you will be having this thing about it. So what happens is this nasal spray is very this thing by the medicine practitioners because the delivery mechanism is a bit different and nobody has ever used vitamin D as a nasal spray and it works like wonder. Okay, the bioavailability becomes very good. So before taking nasal spray, you do vitamin D testing of your serum level. It will be very less. So after 1 month of treatment, you will see that optimum range of vitamin D is there in your serum. Understood. So this is how we also stabilize drugs by putting it into a liposome. Liposome is nothing but a lipid, like a fat droplet. Inside the droplet, there is the medicine. Okay, and you make it into a mist or aerosol, which will enter into your nasal cavity and it will get dissolved. Okay. So this is how we'll prepare this thing. This what do you call is liposomes in the lab. So long chain fatty acid esters with glycerol and then make it rapid vibration so that it becomes like. So there are some this thing also available in the market. Some kids toys like this. You can make it as your paperweight kind of. So there is immiscible liquids. One is blue color, deep blue, and another is like watercolor. In between there is a like several other things moving. So if you shake it, it becomes like completely blue, and after some time again it becomes blue and colorless kind of. So this is also the same kind of strategy that we do, and eventually we'll able to stabilize the peptide by using things like this that we call it entrapment, covalent bonding, absorption, encapsulation. These are all common things for you as well. And therefore, I have put it. And these are again some strategy to preserve your active site. Okay, so you can use several strategies so that your active site remains what we call is hidden from the exterior, and thereby you enhance the what we call function of your what we call is peptide based therapeutics. So this was our paper that we reported like 4-5 years back when we isolated the what we call is bioactive peptide. Okay, so we analyzed it, we understood its bioactivity, and we reported nicely, and we were very happy. But the happiness ended in smoke when we eventually found out that the stability of the peptide is very low. So what we can do about it? So we were doing a lot of brainstorming, and there was no scope to enhance the stability because the molecular weight is already very low. As you can see, 9 amino acids long, and the molecular weight is 11 Dalton kind of 11 to 14, 15 Dalton. As you can see, the later on the bottom lines of these SDS page bands, these are very thin. So it was very difficult to handle, but we were very much sure about the bioactivity that it was being demonstrating repeatedly. When it was like every time we do the test, every time we are getting excellent results. But the problem was stability. So what we can do? So we eventually started a lot of brainstorming, and eventually we came to that by peptide assembly, by artificially enhancing the what you call overall molecular weight of the peptide, we can definitely able to enhance the stability of the peptide. And therefore we tried a lot and eventually we succeed to see it, and then one more problem appeared, one after the another. Small labs like us have to deal with a lot of this. So what has happened is we found out the way out. Okay, so we identified a peptide which was very good, but the stability was very low. And then we also found out the उसका तौर भी मिल गया, मतलब उसको कैसे enhance करना है वो समझ में भी आ गया, सबसे बात किया and did everything, and eventually found out that if we artificially enhance the molecular weight by putting it into an artificial scaffold, and then we can able to do it. So in order to do the scaffolding also, we were not very sure. So we needed to be a foolproof proof that okay, after doing this, we can definitely able to stabilize the peptide. So we needed to depend on a lot more on what you call is simulations. So other than simulations, what happens is your false positive and false negative rates become very high, and therefore you eventually cannot able to economically do your experiments. Every time you do a wet lab experiment, you need to invest a lot in your reagents, your equipments, and so on and so forth. So if you do a wrong experiment and you eventually end up with your losing your fund because you have already exhausted all your reagents. We could not take any chances because we are a very small entity. And therefore we looked into other matters, that how much simulation, how much scaffolding we should do to make it stabilized. So we eventually looked into this thing, and we had our share of this thing also. Moral boost when we had an interaction with this particular lab of Cavendish lab. Now this lab is also very famous because during 2012, please remember, boys and girls, ladies and gentlemen, that during 2012, this lab has proposed that there is a significant risk from the coronavirus. At that point of time, we were not talking about COVID-19. So that was like COVID only, coronavirus. Okay. So this coronavirus was a threat. They have identified it, and that particular time only they proposed that there should be a nanomaterial based vaccine which will able to eradicate the pathogenic strains of coronavirus. So eventually it was not happened, and nobody was serious about it. And today we are having this session online that we could have been doing it offline as well. So this is the lab which has done excellent study on peptide scaffolding and multiple different usage to it. So we are not going to those details today. But the peptide this stability concept is also very important, isn't it? So we'll look into it. Just one second. My goodness. So what has happened is so what has happened is this is the unstable peptide. Okay, so our objective is to make it stable. How exactly we'll do it? So we will reduce the overall delta G. According to the thermodynamic laws, it says that the entropy or degree of disorder is inversely proportional to delta G content. So if anything which is having more delta G will definitely have less system. So if the delta G is in negative magnitude, you will also have very high stability. Degree of disorder will be low. And therefore, the overall strategy is to lower the delta G by doing so, isn't it? So we will also able to simulate the whole thing of reducing delta G, how exactly you can do it, so that you can do it experiment, isn't it? So we did our this thing. This is a very general concept of thermodynamic principles. We are not going into it. Achha. So here again, so these simulations of peptides is possible today because of a visionary scientist, Professor G.N. Ramachandran. During this post-independence era only, he proposed about Ramachandran with a protein that is called cola, and he was definitely a deserving candidate for Nobel Prize, a direct research scholar under Professor C.V. Raman, but eventually he could not get. But today also the vaccine design is very rapid because of this particular concept of Ramachandran plot. So if a protein is there which is not sticking to the principles of Ramachandran plot, is an unstable protein. Okay, this is the thumb rule. So that thumb rule was proposed by an Indian who definitely deserved a Nobel Prize. But anyway, we are not discussing the thing. This is the peptide that we isolated and we simulated as well. This is the singular peptide, and then this is the duplex. That means two peptide molecules in very close proximity, making a. And this is the tripeptide assembly of this small molecular weight peptide. Okay, so we have also seen that this is the overall energy dynamics that has been calculated by a particular kind of simulation that is called GROMAX. It is also this thing very famous tool for thermodynamic analysis of material science and then biomedical research, and so this has also given us a proper idea that scaffolding will definitely enhance the stability of a peptide. Okay. So eventually it has been this thing that it has been found out that scaffolding will definitely enhance the stability. Now these are the molecular motions of different domains present in the and there are some this thing also movements that we have captured from the simulation, and I'll request our coordinator sir to play the second video so that we can see how exactly how it looks to become more stable if it's so how it becomes stable and how it becomes unstable, and how they will look like if it is stable, and how it is look like if it is. So we have captured, it's a picosecond based frame which we can capture and make it into a video. So we have made into a video. So may I request our coordinator to please
Yes,
share
sir,
his
yes.
screen and second video.
The screen is visible. Okay, his screen is visible now. Is screen visible now? Just a minute. Check. Sir, wait a minute. We are sharing the video. Is it visible now?
Yes, sir.
Okay. Dr. Surajit sir, should I play the video? Hello? Dr. Surajit sir, am I audible?
Yes, sir, you are audible.
Dear participants, due to technical issues, sir is joining with us in few minutes. Please stay connected. We'll soon start the session. Dear participants, we are extremely sorry for this delay. It is there is bit network issue at the site of our eminent speaker. So uh, stay connected. We'll begin within 5 minutes.
Okay, am I audible, madam?
Yes, sir.
Okay, okay. Now, see, I'm thankful to the coordinator that exact video that you wanted. Okay, so there are two videos. We'll one after another. In the first video, if you see, I see that the protein is highly unstable. So how you will find it that it is highly unstable? Because you can see only the residues of the whole peptide. So, sir, please, this thing in mute condition, if you play it, so I think they'll able to understand.
Yes, yes, sir.
Is it playing?
Yes, sir, it is playing.
My goodness, from my end, it is okay, okay. Yes, yes. So, as you can see, sir, madam, that the peptide is highly unstable and you can only see the residues, not the complete thing, but some of the most stable parts of the peptide, because we have put it into a simulation box where we are replicating the forces of nature. All kinds of forces that are present in the simulation box is rapidly degrading the peptide. So this is our 9 amino acid long peptide, which is excellent in its work, but stability is very, very low. Okay, so this is the first video. Okay, so later on when we did a what do you call is what do you call is this assembling, that means more and more peptides all assembled in a particular fashion and making it into a scaffold. So when the scaffolding effect was there, if you see the second video, sir, would you please play the second video? In the second video, what you will see is the stability has enhanced to a significant extent. So how you will understand in a visual context that the stability is enhancing? Because you will see that this thing has happened. What do you call is this? What do you call is the other parts of the peptide is also very busy. Okay. So how the other peptide will look like? It will have your this thing. What do you call is the secondary aspect domains? Can you see the what do you call is helix-like patterns? Isn't it? The older time analog telephones were there. So these are the helix-like domains of the protein, which is visible in the second simulation. But in the first simulation, what has happened? Everything did, and everything got torn into small parts, and nothing was visible. Isn't it? Why? Because the stability was also very low. So we used the same simulation, but with a different kind of peptide, and we eventually found out that stability of the peptide has been enhanced. Okay, so this is how it's a visual representation of the whole scenario. Okay, so we'll go to the presentation part. No, not exactly. Okay. Hmm. So it has been shown that the stability has been achieved by using this thing. What do you call as this different modes. So one more application is there through which you will introduce a mutation in the protein and thereby stabilize overall this thing outcome of the protein. So see, this is a very good, nice representation by some other worker. It is not done in our case. So as you can see, there are two kinds of candies. Okay. So as you can see, the previous candy was like this, a blue red candy. Okay, but your anticipated goal is to make candy completely blue and completely red. Am I audible, madam? Okay.
Yes, sir.
Yes.
So what happens is, it is our anticipated goal to make the candy completely blue or red. But eventually what happens, since you are not having a very good enzyme with you, you end up with something like in the right side, as you can see, that the candy has been broken down into several small parts. So that you did. But your anticipated goal is an excellent candy with a proper uniform texture, taste and size and everything else. So how do you do it? Because everything you will do with the help of your enzymes. So what you should do is you will modify the characteristics of your enzymes, which will help you to ultimately get your anticipated goal, isn't it? In order to do so, what you will do, you will remodel your enzyme altogether. We will give some examples of here, how to change the appearance of a therapeutic peptide and stabilizing it significantly. So in order to do so, what exactly you can do? So we'll have some very good examples there. These are some very good examples reported by somebody else where they have introduced a particular mutation and did wonderful things with their peptides. Okay, so I'm not going to much detail. So there are some interesting observations which is visually very catchy. So initially, if you see the wild type PDC is pyruvate decarboxylase is an enzyme which will add a particular kind of proton into a residue, ultimately producing acetaldehyde. But after producing a mutant variety, like mutant is very common today due to the this thing COVID pandemic, everybody knows about mutants. So this mutant, what it can do is it can, instead of a proton, a hydrogen atom, a hydrogen isotope, instead of a hydrogen, it can now accommodate a benzaldehyde, a whole lot bigger molecule to it, and therefore a completely new product you can, isn't it? So likewise, protein engineering can give you excellent results if you can regulate the whole strategy in a particular way. So this is again a very good example. Previously in the wild enzyme, you only needed to add phosphorylated products. For example, see here, D-fructose 6, okay, D-erythrose 4-phosphate. Eventually, you will get a product which is also phosphorylated. But after steps of mutations, you are ending up with a protein or an enzyme in which you can use D-fructose, a dephosphorylated substrate, to make a dephosphorylated product. Understood. Previously, you could not get dihydroxyacetone. Okay, you can get dihydroxyacetone phosphate, but this time you can get dihydroxyacetone because you have introduced a particular kind of engineering or mutation in the protein. Okay, so these are various things that we can able to discuss with some specific interest to this particular thing. So here again, previously in the wild type, you can see the wild type on the, above the arrow there is written wild type. So wild type what? Wild type protein or wild type enzyme. Now this wild type enzyme can accommodate 3-malonyl-CoA. Now 3-malonyl-CoA is a reactant. But if you introduce a mutation and we change the protein structure and we make a mutant, the bottom one, the last one, the arrow which is towards the bottom side, it's a mutant. Now this mutant can accommodate 2-malonyl-CoA. Are you getting it? 3-malonyl-CoA means an extra methyl group was there. Now this time, can you see the chain, the long chain has been reduced to 2. Previously it was in the top side. Can you see? So this is how protein engineering can do wonders. Okay. So here the active site pocket was large and therefore you needed to only accommodate 3-malonyl-CoA. But after the mutation, the structure of the active site, can you see? The structure of the active site has been reduced and changed, and therefore you can accommodate 2-malonyl-CoA. So this is how you change the whole geometry of a protein, and it can do wonders. So there are different strategies, that we do in our molecular biology lab and genetic engineering lab, PCR based methods to introduce mutations. There are specific things, examples where they have done excellent results. Some other labs, then we have used a lot more strategy to do a lot of stuff to understand that how exactly machine learning can help you to generate mutants, okay, and to have to rule out false positives and false negatives. So if you are still connected and if you are interested, we'll go to a particular kind of what do you call is where we'll use a particular kind of strategy to understand that how exactly machine learning works, okay. And it has been useful for COVID vaccine development. These are a lot more things they are to analyze the mutants will look like and to analyze their property. And eventually in 2020 we reported the whole report on this COVID-19, and we are thankful that we eventually ended up writing a paper which has been also showcased by WHO, and in that study we have reported a lot of use of machine learning tools and techniques for vaccine research and development. Okay, so there are some limitations even for vaccine research, kind of related protein design and protein therapeutics assembly, such things by using computational tools. There are limitations as well as scope. Okay, so now all these things is being possible because of whole genome sequencing techniques that has been developed very recently and now been extensively used by all biomedical labs. For example, for COVID-19 also, a lot of genome study has been reported, and these are very useful, extremely useful. And the strategies are like this. I'm not going to discuss them. And this is the whole genome shotgun sequence of a particular organism, a small one, a bacteria. And this is how a whole genome sequence looks like in a database when you are using computational. And we do a lot more hands-on even, we'll do it, but after I complete all the slides. So this is one more thing that we did during 2014-15. We reported it, but started 4 years back, approximately 2010, and we got a little funding from an external NGO which is looking for a cure for a rare genetic disease called birth syndrome. They were interested to explore what is the cause of it, and we eventually were able to identify the cause of it and reported the result. And it is also again, we are thankful that it has also been this thing showcased by this particular NGO. Where we used a lot of machine learning tool, which was that time and was very costly and inaffordable for us. And later on, we thought that it is very difficult to explore this kind of costly things, and therefore we developed our in-house strategy and mechanism. And eventually, in 2020-21, we filed the first patent from our lab, and eventually we are also in talk with an MSME, a medium-size industry, to monetize our product to certain parts. And we are hopeful that we will able to. Am I still connected, madam, sir?
Yes, you are connected.
Okay. So what has happened is, and we are really looking forward to it to monetize the thing that we have developed in our lab, because there are some shortcomings and many this remains inaffordable. And if it is affordable, you can run it because you are running short of your hardware. So these are the things that we anticipated to develop, and now we'll again. May I request? Oh, is there any fourth video, coordinator, sir?
No, sir. 3 videos.
There is no fourth video. We have sent only the 3 videos. Okay, fine. So what we'll do is we start using the simulation toolbox. So would you please share with us the first link? We'll do a little simulation. I'll give a little introduction to it, and then we'll try to do the simulation. Okay. So, sir, you can share your screen, and the third link that we have, you can share in the chat box, as well as you can click on it and then share your screen.
Third link of YouTube, no? Or is different?
No, third link of simulation, I think. Fourth link. And overall, in the email, if you see, there is a fourth link, which is not a video, but a link to a database. So we'll enter into the database and we'll do a little hands-on.
Okay, sir. Okay.
So for that, yes, for that I can unshare my screen so that it remains. So participants, if you are interested, you can definitely this thing participants. So what has happened is, as I have said, that the recent developments in genomics has given us plenty of opportunity that we use computational tools rapidly for recent research on biomedicine. As you know, that vaccine development was very rapid due to this kind of advancement. That is, so we'll see a little glimpse of, not very detailed, but very short.
So this one is screen, na, sir?
I do not know. No. Is there any other link? Just
So one is for
check
this
the email.
downloading this Rasmol software, and another. So that was the fourth link.
Just I see. Any links for total? I have sent, sir. I'll just check it out. Okay, just hold on. Okay. Just 1 second, sir. I'll just check it out and understand. Okay. Okay, great. Fine. So, sir, okay. So what has happened? I hope many of us has already downloaded and installed. Now this Rasmol is a small package of approximately 25 MB of code, which has been written in Visual C++ and then a little of Python. So it's a very small tool and an excellent tool which can give you a scope to simulate about the stability of peptides. Okay. So in order to do the hands-on, we have shared with you a link for the software, which is free to use, but with limited this thing scope. At the same time, we have also shared with you some two data files. Okay. So now may I request our coordinator to click on the icons. So from the beginning, if you start, so it may be easier for everyone to like do also similarly. Many may already know, but many may find it difficult. So, sir, close the whole thing and then share your desktop. From the desktop, you directly click on the icon that is there in your desktop, so that they can also do the same if they want. So you close this from beginning all over again. So in your desktop, there must be an icon. Yes. From this icon, you please click. Yes. Okay. Okay. And keep the window like this. No, sir. Do not this thing. Maximize it. Keep it like the fine. So, sir, you put it in a corner, and then 1 second click on the same this thing icon. So two windows will come, and two simulations will run side by side, so that they can also understand what is the difference between the stable one and the unstable one. Okay, fine. Thank you. So what has happened here, participants? So the small code we have opened. So if you click on the code, two kinds of window will come. One window which will be completely black, and another window which is white in many times. So white is the this thing what you call is command prompt, and you need to write codes on that. But this window, black window, is GUI, which we call as graphical user interface. So we'll use this because it is much more easier to perform. Okay, and remember. So what has happened is we have opened two panes for two simulations that will run side by side. One is an wild type protein. Please remember, wild type means what? Normally जो मिलता है, ठीक है. सोचिए कि जैसे कि आपका जो इंसुलिन है, जो नॉर्मली आपका बॉडी में बनता है, वही इंसुलिन आप अगर मार्केट करेंगे तो आपको दिक्कत हो जाएगा. क्यों? उसका जो हाफ लाइफ है वो बहुत ही कम होता है. तो आप जब इंसुलिन को निकालेंगे, उसको बोतल में डालेंगे, उसके बाद शेल्फ पे रखेंगे, तो उसके लिए आपको बहुत ही स्टेबल इंसुलिन की जरूरत है. क्योंकि अगर 10 दिन में उसका एक्सपायरी खत्म हो जाता है तो कंपनी को मुनाफा नहीं होगा. तो कंपनी को क्या चाहिए? मुनाफा चाहिए. How you will ensure the munafa for your industry? So by stabilizing it, consider that we are two varieties of insulin. One is the novel one, that is the wild type, and another one is the stable one that you have designed. Okay, so it could be any protein. I'm just giving you an example of insulin because it is very common. So if you want to call some other names, it is also very okay. This particular structure is of a growth factor that we are interested in, and it's a small part of it. We'll do a very small simulation, which will take very small. If the molecular weight is big, your simulation time is also very big, isn't it? क्योंकि आपको बहुत सारा कैलकुलेशन करना पड़ेगा सॉफ्टवेयर, और सॉफ्टवेयर भी बहुत छोटा सा, तो उसको भी टाइम लगेगा. तो हम क्या करेंगे? एक छोटा वाला पोर्शन लेंगे पूरा प्रोटीन का, उसको एनालिसिस करेंगे और उसको एक्सट्रापोलेट कर देंगे. ठीक है, तो चलिए हम करते हैं. तो क्या है? वी आर हैविंग टू विंडोज. From both the window we are having the file. तो हम किसी भी एक का फाइल मेनू में जाएंगे सर. फाइल में क्लिक करेंगे.
हाँ सर, क्या है लेफ्ट में?
तो उसके बाद आपका नो फाइल मेनू में जाइए. हाँ, उसमें ओपन कीजिए. ओपन में जाइए, नीचे जाकर हाँ ओपन कीजिए. तो गेट डाउन एंड क्लोज ओपन. हाँ यस. Okay, now to that data file that you have downloaded already. Okay. So you first click on the first data file. Okay. So it will get open. इसको open कर लीजिए. Again click on the open. Okay, it will open. Okay. Now do not disturb it. Now go to the again do the file and go to the second what do you call data file. Go to the file option. Yes, the second one. Absolutely fine. Okay. Now these are the skeleton of a two protein. Okay. Two different protein, but very similar. So you can ask me, sir, why these are similar? Because these are the part of the same protein only. But in one, we have introduced a mutation through a PCR based method. Now in PCR, what happens is you can manipulate the gene or the DNA. How you can manipulate? To make a PCR product to come, you should put a primer. Now in that primer, we have already introduced the mutation. Now, what is the mutation? For like everybody knows, for example, instead of an adenosine, we have put a thymine. But to find out the exact space, it needs a lot of task to be done. So, for example, let's say that we already know where exactly we have introduced. Okay. So, after everything, now these are the two parts of the protein. One, the wild type, the normal wala insulin. The other one, that you have designed all by yourself. And it's an excellent one because it is having an extended shelf life. ठीक है, तो देखते हैं उसका क्या होता है. तो पहला वाला ये दोनों जो प्रोटीन है, प्रोटीन क्या आपका स्केलेटन दिख रहा है. स्केलेटन मतलब क्या? उसको सी अल्फा बैकबोन. ठीक है, तो सी अल्फा बैकबोन मतलब उसका जो मतलब only the covalent bond is there, that is only showing, that is the peptide bonds. Okay. Now we'll put the atoms on it and it will become complete. So in order to put the atoms on it, may I request our coordinator to go to the what do you call is just one second. Wait. Yes, go to the display window. Yes, go to the display window and put on molecular surfaces. The bottom one, molecular surface. Bottom वाला जो है drop down में, हाँ, इसको click कीजिए. And give a little time. Give a little. Okay. Now we have generated the molecule. Okay. The second one also will do the same. So participants, if you want, you can also go ahead. So in the second window, we will also do the same. So on the skeleton, we'll put the atoms and it will become a molecular substance. Now this is the three-dimensional structure of the enzyme or the protein or whatever you. Okay. So now these two are there. Okay. Can you able to see these are approximately similar in structure, doing everything same. But we are interested in a more stable protein. That is our anticipated goal. Okay. In order to isolate that which one is stable and unstable, we need to use computational tools a lot, and then we'll able to remove the false positive. Okay, what is a false positive? False positive is something which is not really good, but it is showing that it is good. And in order to do this, you need to invest a lot in terms of your what do you call this thing? What do you call reagents, isn't it? एक reagent है जिसका cost बहुत ज्यादा 25,000 का एक 10 ml मिलता है. So आपको 10 बार करना पड़ा गलत एक्सपेरिमेंट तो आपका तो पूरा पैसा खत्म हो जाए, isn't it? So in order to reduce the risk, what you will do is you will do a simulation initially, and then we will go to the what you call is wet lab, isn't it? So how you will understand that which one is more stable? So for that we will go to the color. Sir, please go to the color option in the first window and click on it, and drop down menu will come where you will find temperature. So you'll click on temperature. Okay, you'll do a little calculation, and on the basis of the overall, we'll say, now the color, the pattern has been changed, isn't it? Some area red, some area blue, some area this and that. What exactly is this? It is nothing but the energy landscape of the protein. So in the energy landscape of the protein, what exactly it looks for? It looks for the area which is more stable. It also looks for the area which is less stable. Okay. Now in terms of molecular viewing of this thing simulations, there is a thumb rule. The thumb rule is if something is blue in color, it is more stable. If something is red in color, it is less stable. Now what exactly is less stable? Less stable means delta G is in positive magnitude. Okay. Now more stable means what? Delta G is in negative magnitude. Okay. तो ये वाला प्रोटीन जो है देखिए ये वाइल्ड टाइप वाला है तो किधर थोड़ा ज्यादा स्टेबिलिटी किधर थोड़ा कम स्टेबिलिटी so you cannot depend on it, you need something which is highly stable. So we'll go to the next one and we'll do the yes small this thing temperature is also this thing for we call it B factor analysis. Okay? So what we'll have is we'll have the now here you see both the protein looks like same. These are in fact same proteins. A single mutation is there in the protein, and therefore the overall stability of the peptide has been rapidly changed. Can you see? We use the same software, we did everything same, but the stability wise it is very different. Completely blue, and some area is red. Red area we can cleave it, we can cut it down. We are having some specific tools through which we can, like it is also there in your body. We call it post translational modifications, PTM. Okay, so PTM we can also do in vitro. Getting my point? So this, therefore, what will happen is we eventually end up with something which is more stable, and it will give you very good results, isn't it? So this is we'll do a little hands-on simulation which will help you to appreciate that what can be done. Yes, that can be done in protein engineering, isn't it? And now if it is pharmaceutically an important, it will explore you, it will give you a scope to monetize on this particular product, and you will get wonderful results, isn't it? If you can generate a COVID vaccine which is stable even at room temperature, the industry or the company will get two like Nobel Prizes. One for finding out a cure for the thing, and the second for the rapidly decreasing the cost. The important cost that is borne this this thing for COVID vaccination is for this thing, handling, storage, this and that, isn't it? So why is this a problem? Because the stability is very less, isn't it? So you can now ask me, sir, we can mutations anywhere and you can see whether it is good or bad. So eventually you can also destroy the protein altogether, isn't it? Your stability is very high. So in body, in human body also, at molecular level, this mechanism is also, for example, due to some error during translation, what there are some errors that can be present. So during that error, you can also generate a protein which is very stable. so stable that it cannot participate in this kind of proteins. We call them inclusion bodies. Okay, so there are some methodologies, for example, apoptosis and programmed cell death and this and that, through which we can eradicate this kind of abnormal proteins which are of no use, isn't it? Like there is one more disease also where you generate a lot of very stable proteins, isn't it? We call it this multiple sclerosis, where you generate a lot of myeloid lesions on your brain and several other parts. So there are several aspects of this protein engineering that we can look into, and we are as a lab who have a specific interest to it. We are looking for collaborators and labs which have excellent computational resources, pharmaceutical tools, and we are very much open to collaborations and outreach activities. So by saying so, I would conclude my session, and if there are some questions from the participants, I'll be very happy if I can able to answer.
Thank you, sir.
And I'm thankful to the court that she has given an opportunity to interact.
Yeah, sir. Thank you. Thank you so much for your valuable talk. Now the session is open for discussion. I request all the participants to unmute yourself and can ask your questions directly, or you can paste your questions in the WhatsApp group.
उद्या मी फक्त 12 वाजेपर्यंत त्याच्यानंतर मला इन्स्पेक्शन.
Hello, sir.
Madam, yes.
Sir. We have received one question from Pishwikar, sir. How do you see use of peptide therapeutics in personalizing the mode of treatment in patients?
Yes. Very right, sir. That you have pointed as you will also know that what has happened. You have taken consider that that you have taken both the jabs of your vaccines. Okay, schedule 1, schedule 2 vaccines you have, and you are traveling to some Southeast Asian countries, for example, Singapore. So if you, when you get down at Singapore, you all over again you are tested COVID and you are COVID positive, and you are like heavily with the symptoms as well. So you'll be amazed that I have taken 2 vaccines instead of that, and recently it is not very old also, and my immunity is also very good, and this and instead of that I'm getting one more time COVID. Why? This is wrong or something? No, that is true. Okay. Yeah. Despite of taken 2 vaccines, both the 2 jabs and not missing anything, you can definitely also encounter with a situation where you are COVID positive. Why? Because the COVID is an RNA virus, as you all know, and it is constantly changing its structure. That also everyone knows. So this gives us an opportunity to explore the thing you have said. Personalization of medicine is a need for the hour. Like let me tell you, like after 20 years, not 20, let's say within 10 years, personalization of medicine will be a common thing, because besides that, you cannot get rid of a disease, because of the genetic makeup is such that the virus is also changing rapidly to cope with the challenges that it is facing. It is kind of a prey-predator kind of relationship. For example, the DR is running, okay, and at the or DR को जो भगा रहा है वो है, so nature will do evolution for both of them, for the DR as well and for this thing also, the tiger also. The deer will get rid of the bigger horns that it had previously, and therefore it will not get entrapped into the jungle due to the large horns it had. At the same time, the tiger will also acquire some ability to run for a longer period of time and for a very this thing with a high pace. Therefore, what happens? Evolution is helping both, and therefore evolution is also promoting both of them to sustain. Nobody will be eradicated. So the virus is also doing the same. Every time you boost up your immune system by putting a vaccine, it changes its structure, and it is also very this thing for this RNA virus. So this COVID-19 for approximately it is 20 years old, and HIV is like 50 years old. So we have done nothing for HIV. How can you expect that you will all over in a day will eradicate COVID? It is also an RNA virus, and it is also rapidly changing. Eventually, you cannot able to do it completely, and therefore the personalization need is there. For example, you have taken a vaccine for this thing, but not for the Singapore variety, not for the alpha. So what you need a battery of vaccines. It is not that you will take 10 ml of vaccine altogether. It is not that you will just take 1 ml only, but in 1 ml it will be kind of like I do not know what is the strategy tomorrow, but there should be which will protect you from all possible threats as well, and then only your vaccination will be completely successful. Like polio. Now in polio, you take the 2 jabs and you are completely eradicated. Why? Because polio, it is a DNA vaccine. It is highly stable and it is not rapidly changing. COVID-19 is changing, isn't it? So as you have asked, you are absolutely right that we are definitely looking forward to a period when we will desperately need personalization of medicine. A single medicine will not work for everybody. Today also we are doing personalization. For example, you take paracetamol 600, but your kid will take 200 ml. That also a suspension, isn't it? So here also there, but this thing is very low. The how shall I say? The intensity of the personalization is small. Tomorrow it will be much bigger. So you are absolutely true that we are looking into a situation where large scale personalization is definitely needed. So this is how I would.
Thank you so much, sir.
I am also thankful to the participant that some of us are still sticking around and everybody has not left.
Hello, sir. I would like to ask
Yes,
question.
madam.
That is, how can we increase the stability of peptide therapeutics using machine learning?
Yes, madam. As I have demonstrated, we have demonstrated a tool where you are making a peptide to learn that how a peptide will look like when stability increases. So machine learning is a thing. I mean, I have demonstrated only the analytical part, not the application. I mean, how exactly you will do it, because the scope is also very limited. But we have shown you a way. Machine learning is nothing but your learning only. The way we learn things, we have our experience and then we take a decision. So before developing the software, what the software people did, and those people who wrote the software, they made the software learn that if they will calculate each and every delta G for each and every atom. Understood? So if the delta G of each and every atom is within a certain limit, it will become blue in color. So this is machine learning. You are teaching your machine to generate blue color when delta G is less than point. Are you getting my point, madam? Now, if it is more than 0.5, it will be orange in color. So you are giving an instruction. Like when you are like clicking an icon, Sar is clicking an icon that I have shown. When he's clicking an icon, he's giving the computer an instruction that when I'll double click it, you open a window where in the window these many toolboxes will be there. So this is a kind of this is the simplest concept of machine learning, how machine learning works. Now, how you make use of it, you need to have FDPs like 5 more FDPs like this to understand. Because we do not belong to this thing. I also have a very limited understanding and limited knowledge. But since I am having a specific interest, therefore I can able to simplify this kind of concepts to you. But I'm sure if you stick to this and study a little more, you will also able to understand yourself that how you can make use of machine for your kind of work you want to do and the research you want to do. So we can definitely able to use machine learning for protein engineering, as I have said, to introduce mutations and then to analyze what is the effect of the mutation. So अल्टीमेट गोल इज व्हाट, मतलब आपका अल्टीमेट गोल है ये कि आप मतलब क्या करें, आपको लैब में कुछ करने से पहले कंप्यूटर में आप एक बार देख लीजिए जो मतलब अगर ऐसा करेंगे तो ठीक रहेगा कि नहीं ठीक रहेगा, ठीक है ना। फॉर एग्जांपल यू हैव इंट्रोड्यूस्ड ए म्यूटेशन, दैट मींस यू हैव रिमूव एन एडेनोसिन इंस्टेड ऑफ थाइमिन। ये वाला ये सीक्वेंस में करने से रिजल्ट क्या होगा, ये अगर आपको पहले से चल जाए पता, तो आप क्या करेंगे, इसको करेंगे या नहीं करेंगे। अगर बहुत अच्छा रिजल्ट मिलता है सिमुलेशन में तो जरूर करेंगे, अगर अच्छा नहीं रिजल्ट मिलता तो क्या करेंगे, यू विल नॉट डू दिस, isn't it? So this is how you can use machine learning for most of your kind of work that you want to do. So this is how I would like to this thing explain.
Okay, thank you, sir.
Yes, madam.
Over to you, Vijaya.
Yes, ma'am. Thank you so much, sir, for your informative session and showing us the live demonstration of hands-on simulation. Now I request Mr. Vishal Thorat, sir, to sum up today's session.
Good evening, one and all. I, Mr. Vishal Thorat, here to present the sum up for today's session delivered by Dr. Surajit Debnath, sir. First of all, I thanks to Dr. N.M. Bhatia, madam, coordinator, for giving me the opportunity to sum up this session. So we talk about the session. It was excellent and informative, so that we are helpful. First of all, the thanks to Dr. Debnath, sir, about this today's session. Sir has focused and highlighted on the very important aspects like about diagnosis, then therapy, as well as drug delivery, and one of the interesting parameter, the engineered antibodies, and as well as the preclinical and clinical studies clearly mentioned, and under that the consideration for peptide-based therapeutics. Sir has also guided about the how to stabilize peptide therapeutics, preserving therapeutic protein and suitable methods, then alternating reaction specificity through saturation mutagenesis. Sir also discuss about the structural alternative activities and recent development, recent process. Then finally, Sir has discussed the hands-on training about to enhance the stability by using the learning Rasmol model domain. Once again, thank you very much, sir. The session was very interesting, informative, which definitely help us in academics as well as those who are participate. The session is a research work. I will see you in future coming sessions. Thank you. Thank you very much, sir.
Thank you, sir. Thanks a lot.
Thank you, sir.
Thanks to Dr. Debnath. Sir, is it possible for you to put your camera on so that we can have some of the pics?
Yes.
Yeah. Over to you, Vijaya.
Yes, ma'am. I, Ms. Vijaya Saugule, on behalf of Bharati Vidyapit College of Pharmacy, Kolhapur, takes this opportunity to thank our eminent speaker for his wonderful presentation and valuable time. Thank you so much, sir. Now I request all the participants
Thank you.
To turn on your camera so that we can have photo with our eminent speaker.
कहाँ चले सर?
Okay, it's done. Thank you, everyone. I would like to announce that today's session for day 3 are concluded and the feedback links will be shared on your WhatsApp group shortly. I request all the participants to join tomorrow using same link 10 minutes before the actual time for ATAL sponsored FDP on pharmaceutical quality system for product lifecycle management organized by Bharatiya Vidyapeeth College of Pharmacy, Kolhapur. Thank you everyone.